I'm Yuji Miyahara at TMDU. I have been studying in the field between nanoelectronics and biotechnology in collaboration with Dr. Miyuki Tabata for the purpose of detection and analysis of biomolecular recognition and cell functions in a parallel manner. We are very much interested in investigating various kinds of signal transduction mechanism from biomolecular recognition to electrical signal for solid-state biosensors. In 1990, Human Genome Project started and production of biological information drastically increased. So high-throughput analysis and data processing have been required, and great progress have been made in the research field of bioinformatics and high-density chips. Typical example is a biosensor based on field effect transistors, FET. A field effect transistor is a fundamental device in a large-scale integrated circuit, LSI, and has several advantages such as miniaturization, high-density integration of the sensing areas, and integration of signal processing circuit for the functional analysis on chip. So it is possible to access single cells in a parallel way. In most of the solid-state biosensors, biomolecules are introduced on the surface of the sensing areas of the transistors. Typical example of the biosensor that we investigated is the sensor for DNA analysis. In this case, oligonucleotide probes are immobilized on the surface of the gate of the ion-sensitive field effect transistor, and a single base extension reaction is designed in combination with enzyme DNA polymerase on the surface of the transistors. As a result of single base extension reaction, negative charges based on phosphate group increases and the protons are released. If we measure the increased negative charges or protons, we can recognize the event of single base synthesis on the transistors. We have licensed the patent to the venture company, and this technology has commercialized as a proton DNA sequencer. We can detect not only biomolecule but also cell functions using biotransistor. A novel detection system for detecting multiple antigens expressed on the surface of the cells have been proposed using ion-sensitive field effect transistors, combined with enzymatic chemical signal amplification. In this system, cells are captured on the surface of the FET non-specifically, so that the cells to be analyzed are independent of the expression of specific antigen. Antibodies conjugated with enzyme are then bound to surface of antigen specifically to characterize phenotype of the cells. Upon addition of the substrate for the enzyme, local pH changes are generated on the surface of the FET depending on the expression level of the surface antigen. If multiple antibodies conjugated with different enzymes are bound to the surface antigen, multiple antigen could be detected by sequential addition of the respective substrate. 4 types of breast cancer cell lines were used as a model of CTC, and epidermal growth factor receptor, EGFR, was used as a target molecules. The expression level of EGFR on the membrane of MM231, MM453, MM468, and BT474 cells were systematically analyzed using the FACS method. This figure shows the FACS data and histogram of the fluorescent intensity for each cell type. The fluorescence intensity of MM468 was highest among the 4 cell lines, and MM453 showed the lowest intensity. This trend is consistent with the result reported in the literature. Each of 4 cell lines was seeded on the surface of the FETs, and anti-EGFR antibodies and secondary antibody were sequentially applied to the cell surface. Then 10 millimolar glucose solution was added to the reaction chamber of the cell-based FET. The potential shift 10 minutes after addition of the glucose were compared among 4 cell lines. The amount of potential shift was bigger in the same order as a result of FACS experiment. Comparison of EGFR expression levels measured with ISFET method and FACS method is shown in this figure. A good correlation could be obtained between the pH values measured with the cell-based FETs and the fluorescent intensities measured with the FACS with a correlation coefficient of 0.976. The isolated CTCs are exposed to various biological and physical environments such as shear stress, immune surveillance, cell-to-cell and cell-to-matrix interactions. While in circulations, membrane proteins may change throughout tumor progression. CTCs undergo epithelial mesenchymal transition, EMT, and as a result, expression of epithelial markers including EpCAM decrease, and the expression of mesenchymal markers such as vimentin increase. Since the current method is based on epithelial markers, the capture and the detection of epithelial marker negative CTCs would be limited due to downregulation of epithelial marker during EMT. On the other hand, we can use several kinds of enzymes with the proposed method. When those enzymes are conjugated with antibodies recognizing epithelial antigens or mesenchymal antigens, it would be possible to detect the different surface antigens simultaneously by sequential addition of the respective substrate and to analyze the heterogeneity of the cells once the cells are captured on the surface of the transistors. Thank you for watching.